Yes, this is where we stopped. And I was telling you that I'll give you such a cell and I task you to identify which phase. Now, there are key features you should look at. Okay, and let me highlight them. Number one, look at how these chromosomes are. Okay, and number two, look at the centromeres. Okay, and sometimes the nucleus. But the two key features is the arrangement of these chromosomes and the centromeres, but mainly the chromosomes. Still, let me insist on that. Mainly chromosomes, because I'll give you, for example, I'll say which phase. I'll give you a cell and I say identify which phase, and I give prophase 1, prophase 1, and then I say prophase. Okay, and then I say metaphase, and finally anaphase. Now, if you don't know this, then you will confuse between these two answers. Is it prophase 1 or prophase? Now, prophase 1, this is the prophase of meiosis 1. And we shall reach there and see actually how it is. But prophase, this is actually normal prophase, what we are looking at. Okay? A cell that is in mitosis, normal mitosis, that is prior to cell division. Now, you should identify that. Now, after a cell has done all this, the centromere, the centrioles, they are migrating, the nucleus is shrinking and disappearing, the nucleus the same. It will finally, it look like this in a late prophase. And what's up with late prophase? Like I told you, this level, O level, we don't have late and I. Okay? Can we mute? Who is speaking? No one? Okay. If you want to say something, just raise up your arm. And yeah, I'll pick you up. Or you can use the chat box. I'll be able to read your concern. Question, a supplement, a disagreement, you are free to disagree. Okay? Because it's a discussion. Okay? It's a discussion. And I should try enough to convince you. If you fail to be convinced, then you tell me. I would use other means to convince you. So chromosomes, they shorten and become thin as two chromatids joined at the centromere. Then the nucleus disappears. Now, what is a nucleus? All of you are conversant. All of you, you are free with a nucleus, but few or no one here knows what a nucleus is. This is a cell membrane, and this is a cytoplasm, and this is a nucleus. Oh, sorry. The brains cannot cross. So this is a nucleus. Now, inside the nucleus, there is another dense structure. And that dense structure is the one that we call a nucleus. So the nucleus, together with the nucleus, they completely disappear in this phase of mitotic cell division. Why do they disappear? So that you create a room, for the chromosomes to successfully divide. Okay? Sorry, separated, not divide. They divide they replicate in phase. So here they successfully separate so that each cell that is formed has the exact copy, okay, of the number of chromosomes. Then the nuclear membrane breaks and the cell and it disappears. And you prepare. Now this cell, this is really red prophase, okay? The cell that is really transiting into another phase of mitotic cell division. So let's actually see what happens in another phase. Let's actually see what happens in another phase. And by the way, can you guess another phase? Which phase? Which phase? Yes, yes, which phase? Yes, I hope you've guessed right. Now, the preceding phase is the metaphase. And this one is the easiest to understand. It is the easiest to understand. And by the way, you should not confuse metaphase of mitosis and metaphase of meiosis. Those two things are different, completely different. Okay? Completely different. And we are going to see, we shall really appreciate how different those two phases are. Now with metaphase here, you know, my cell, the aim is to divide. Okay? The aim is to divide and you form two daughter cells, and each of the two daughter cells must have the same number of chromosomes as the parent cell. So in other words, remember there are two cells. Sorry, it's one single cell that is divided. So there must be mechanisms that is employed to separate the two chromosomes. Maybe if I'm to take you back, why chromosomes? Of all things, why chromosomes? Why are we talking about chromosomes? What's that special thing about chromosomes? Okay. Now, if I'm to take you back, we said or we agreed that these chromosomes, they are actually the ones that bear DNA. You remember that? And we said that this DNA is actually a genetic material. It's a genetic material. What does that mean? If you remember the first lesson we had concerning genetics, I told you that you guys, you are what you are because of your DNA, right? Yes, everything about you. Just look at yourself. You might be fat, you might be tiny, you might be brown, light-skinned. Whatever is about you is because of your DNA. So meaning, any tampering, if you tamper with your DNA, then it means you tamper with yourself, with the entire organism. Okay? And yes, we agree that DNA is wrapped to form chromosomes. And now these are the chromosomes that we are separating. And remember, if I draw, remember in interphase, our DNA has to divide. The chromosomes must divide. And so therefore, they are going to arrange with metaphase, they must arrange on the equatorial, okay, before it is not yet, it's not yet. we shall find it. So the spindle fibers are formed. Now, what are spindle fibers? Now, these are macrotubules, or these are like fibers. It's like you get a rope, okay? You get a rope, you tie it here and here, here and here, and you start to pull. You guys, you remember tug of war? Have you ever been in a tug of war where you have people here, okay? And another group of people here, and the two are pulling a rope. I remember the last tug of war I involved myself in. We pulled this rope until when it torn into pieces. And each group had its own pieces. Its own piece, in fact, not pieces. So it was go. We were energetic and they were also energetic. I think I was the one who was energetic in my group. So the same applies to this. So these are like ropes, okay, that are attached on the centrioles, okay? They are attached on the centrioles and they pull, okay? This one pulls and this one also pulls. Now, as this is pulling and this one is also pulling, you create pressure on the centromere and the centromere will end up separating. Okay? Yes. Let's enjoy. So here spindle fibers are formed in metaphase, and the two spindle fibers, they do radiate from the two opposite poles, and the poles have a centriole. Okay? Yes. Then on top of spindle fibers forming, this is the main point as far as metaphase is concerned. This is the main point, that the chromosomes, they do arrange themselves at the center of the spindle or equatorial plane. This is the main point. Yeah, you have a look at this. This is what I'm talking about. If I give you such a drawing, I'll give you such a drawing and I'd be like, which stage of cell division is shown in the figure below? And I say A, I put prophase. I put B, I put anaphase. I put C, I put maybe Pterophase. And then finally D, I put none of the above. Can you drop your answers? And you should do that in just 1 minute. Hurry up. Hurry up, guys. What would be the answer in this case? Yes, that's brilliant work, I can see. Surprisingly, again, you guys have done it. You've all given me the same answer. All of you, you've posted the same answer. Now the question is the same answer correct or the same answer is not correct? But yeah, all of you given D as your answer, and I can say brilliant work. The correct answer here is D, none of the above. So if I give such a diagram, look at how chromosomes are arranged. That's the main key word. Okay? You remember I told you in prophase, look at how chromosomes are arranged. And by the way, this is metaphase and metaphase 1 of meiosis. All they do arrange themselves in this manner, but still they are different, and you will notice how different these two phases are. But if I give you such, and you see your chromosomes, in fact, these are chromatids, they are arranged on the equatorial plane. You see this? In such a manner that if this one pulls towards this side and this one pulls this side, then the chromosome is going to separate into two chromatids, and the two chromatids will move to the opposite poles. Now, you see why I want you guys to know these terms. Now I'm saying chromatid, chromosome, spindle fibers, poles. So I hope you know the difference between a chromatid and a chromosome. Maybe if I'm to remind you again, if I draw this, okay, the whole of this is a chromosome. Okay? But if I simply draw this half of the chromosome, then that's what becomes a chromatid. So you don't need to get confused if I say chromatids separate, chromosomes separate. And actually those words are key as far as distinguishing between meiosis 1 and mitosis is concerned. Not so? Yes. Then finally, towards late metaphase, our spindle fibers begin to contract and shorten a bit. Okay? Shorten a bit, separating sister chromatids of each chromosome slightly at the centromere. But this is mainly late prophase as the cell is transiting to another phase. Which phase? Can guess with me, guess. Yes, you've guessed right. The phase is Anaphase. And let's see what actually happens during Anaphase. Now with Anaphase here, this is where you're going to separate your two chromosomes stroke chromatids, depending on which phase you are dealing with. So here, the sister chromatids, you remember what we talked about sister chromatids? What are they? Why aren't they brother chromatids, but sister chromatids? Yes, we said that chromatids, sister chromatids, they are chromatids of the same chromosome. So if this is, for instance, the whole of this, if this is chromosome A, then this chromatid and this chromatid, they are referred to as the sister chromatids. So if I say that sister chromatids of each chromosome begin to move, they separate and begin to move to opposite poles, it means that the spindle fibers, they will shorten, they will contract and shorten, they are attached on the chromosome at this point known as centromere. There are specific protein molecules that are responsible for attaching these chromosomes on the centromere. And those protein molecules are known as kinetochores. So if you want, you can find out more. If you don't want, it's okay. No one is going to ask you that. So now, if this spindle fiber, have a look at this. This one is attached from here and this one is also attached from here. So they are playing a tug of war. This one is pulling towards this side, and this one is also pulling towards this side. So they pull until when they create strain on the centromere, and boom, it separates so that this chromatid goes towards this side, and this one also goes towards that side. So they are the sister chromatids of the same chromosome that separate. Okay, guys, and they do separate, and they do move to the opposite poles in this anaphase. Okay? Yes, that's brilliant work. Then the movement is facilitated by the spindle fibers, by contraction and shortening. Now, if I'm take you back, you remember I told you that in interphase, that's where we shall synthesize more ATP. And if ATP is not enough, then the cell cannot proceed to the next division. What does that imply? If you don't synthesize enough ATP, then you still go back until when ATP is enough and you are allowed to proceed to the next stage. So likewise, now, before that, likewise. Now, think about what's the ATP for? Do we use energy in cell division? And if we use it, where do we use it from? Which phase? And what is it for? What does it do? Now, the answer to all those questions is one. It's this. For these spindle fibers to pull, to draw chromosomes away from the center, away from the equatorial plane, away from the spindle plane, the spindle fibers must contract. And you know, when spindle fibers contract, they will shorten. Somebody has a question? Pavin, if you want to ask, please feel free to raise your arm and ask. So these spindle fibers, they will contract and shorten. And when they shorten, they pull the chromatids away, hence weakening the centromere, and the centromere will separate. And so that process of contracting, it's an energy consuming process. Really an energy consuming process. So therefore, you must have enough ATP to allow that process to occur with ease. Anyway, let's see what are we talking about. This is exactly what I'm talking about. Okay? And the two chromatids, you see, that made the sister the chromosome. The two sister chromatids, this one and this one, that made the chromosome, they are now separating. I think, and each is being taken to the opposite pole, to this one and this one. Now, why are you doing all that? So that after this chromosome, these chromatids have reached the opposite poles, then you come and okay, you come and divide this cell from here so that you form this cell with all the chromosomes, and you form another one with all the chromosomes. Okay? So if I give you this and I be like, guys, which stage of cell division is this? Okay? And which stage of cell division is this? And I put number 1. So I put A, anaphase, anaphase 1, I put B, metaphase 1. Metaphase 1. I put C, prophase. And finally, I put D as anaphase. So what would be your answer? This is anaphase. This is a P. Yes, drop your answers. Chap, 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 chap. Let me change settings so that you don't cheat me again. I think I did. Yes, the correct answer here is D. And I was astonished that almost 99% gave D. And that was brilliant. Okay? It was brilliant work. And yeah, keep it up. Keep it up. Now, you can guess what may happen in the next phases. Now, here we have another diagram. And this one is red, very red. Here the cell is transiting, okay, to another phase. But trust me, they will give you this, okay? They will give you for metaphase. They will give you. Now we have another phase, which is Pterophase. And you can guess what happens here. You can guess. It's some cool stuff, some easy stuff, okay? Now with the Pterophase, here you are forming now your two cells, your two daughter cells, and everything is resulting from them. And with Pterophase number 1, the chromatids become chromosomes after reaching their opposite poles. Yes. Remember, they are pulled here, they are being pulled, and they will reach the opposite poles around here. And then this one cleaves. And you separate them and you form your two cells. Now, when the chromosomes reach the opposite poles, when the chromatids, reach the opposite poles, they join together at the point known as the centromere. Okay? And then, boom, become chromosomes again. Then the cell membrane begins to constrict so that the cell is divided into two daughter cells. And the constriction is completed still within the same cells. And finally, form your two daughter cells. Okay? The nuclear membrane and the nucleolus, they reappear. Remember, that disappeared in order to create space to pave way for cell division to occur. Now here, you are resorting back. You are trying to form back a normal cell. Spindle fibers disappear. Yes, because they have done their part of putting these chromosomes, of drawing these chromosomes closer to them, to the two opposite poles. And after doing that, they will disappear and you form the two cells. And chromosomes become invisible again. Remember, in interphase, we started with very thin structures known as chromatins. So the chromosomes that were formed prior to division, they now shrink again and become invisible, okay? As long as thin structures, okay? And you cannot see them you can assume, okay, that the cell is not dividing. And that's when now your new cell is going to join the interphase. Okay? And so you can have a glimpse at this. So I can give you this and I say, which phase is this? And you should be in position to tell me which phase. Okay? Yes. So I'll say identify and I give A, Pterophase. I say B. What's my B? I say maybe Anaphase. And then C, I say maybe interphase. And finally D, okay, I say prophase. So which phase? Now, I might give you this or I might give you this, where the cell has completely, it is shrinking to divide. So even if I give you this or I give you this, it is still the same question. So what would be your answer? In just 1 minute,